transfer, and I'm going to talk somewhat about disease progression modelling as well. But before I get to that, a little bit about me and where I am now, what I do. As Peter said, so I've been for just over a year now the head of UCL computer science. So actually, this is kind of exciting. I realised on the way down, this is the first kind of research talk I've given in about a year, because I've been obsessing over, student numbers and is there any future to computer science, and what do we do with undergraduates when they can just program using AI now? So different questions have been kind of playing on my mind for the last year. But at the same time, it's been an amazing transition from being focused on research to suddenly looking across the whole of computer science and thinking about, lots of different research topics and how those interact with teaching and, the rest of the university and the world and so on. So it's been fun. It's not all form-filling and telling people off. There's some nice bits to it as well. UCL computer science, for those that don't know, is an enormous beast. It's the biggest computer science department in England. It's second in the UK, only to Edinburgh, which is similar size. Covers all sorts of different areas. I guess it's quite an applied department, reflecting really, I guess, this environment at UCL, where there's all sorts of different application areas going on that lend themselves to collaboration with computer science. So it's quite a nice position to be in, sitting in that environment where there's lots of people coming to us wanting to do interesting things with computation. I think that really drives the set of research topics that we have here. So the overview, just cut and paste from the website. Anyway, enough about my new life. Previously to being the head of computer science at UCL, I was head of what at the time was called the Centre for Medical Image Computing, and has undergone a kind of rebranding since then under its new leadership into the Hawkes Institute. But what the institute does is, it's been around for about 20 years now, and is a centre focused primarily on medical imaging and medical data and sort of computational techniques for utilising that data in interesting ways. It sits primarily in the engineering school, but reaches out there are several kind of medical faculties at UCL and has very strong links with all of those. So it's become a very important vehicle for translation of basic engineering and computer science innovations into medicine at UCL, and that model, I think, has worked really well over the years. It's grown and grown over that time, and continues, I think, to thrive. So that's really the context of the research that I'll tell you about. So as I said, I'm going to talk about two things. I'll start with this idea of image quality transfer, and I'm going to kind of frame it within the idea of democratization of MRI. So let me start with that idea. What does that mean, first of all? It means just trying to make MRI available to a much broader cross-section of the world. I started working with sort of MRI practitioners in Africa about 10 years ago now, which really sort of gave rise to this line of thinking, at least in my own mind. Lots of other people were thinking about this too. But this map sort of sums it up quite nicely. This is a map of the number of MRI scanners per million people across the countries in the continent of Africa. And what you see very rapidly that is in most of Africa, particularly the central regions, that number is incredibly low, typically 0 or 1 MRI scanners per million people. And you can compare that with, in Europe or China or the US, where that number would be, in the 30s or 40s in most sort of the richest regions. And the presence of machines like MRI scanners really correlates very well with non-communicable diseases and a whole range of different health metrics. Why is that? So there's a sort of obvious financial hurdle of just buying these machines. So that is an obstacle, but actually it's not the biggest obstacle to deploying MRI machines and similar things to these regions of Africa by a long way. So there's once you get over the hurdle of actually buying one of these machines, there's a follow-up hurdle of then maintaining and updating the machine so that they remain usable. And this is a huge problem. As you go and visit hospitals in Africa, this is a not untypical scene where kind of well-meaning charities or external funding bodies stump up some money for a machine and deliver it into some remote hospital in Africa, say, and the machine works for a few months, then something goes wrong with it, and the manufacturer of the machine says, "Yeah, I'm not going there," and so the thing breaks down and just sits there and never gets repaired. And you see these kind of graveyards of equipment where there is no way to salvage these things, so they just sort of cause clutter and are useful. So that's one challenge, is the maintenance and sort of ongoing costs of systems like that. An even bigger challenge is the kind of underlying educational infrastructure. There's not much point putting a sophisticated MRI machine in a hospital in, say, Nigeria, where no one knows how to use it. And at the same time, there's not much point educating groups of people on how to use a sophisticated MRI machine if you can't afford to buy one and maintain it. So there's a kind of chicken and egg situation of having the machines and being able to maintain them, and having people who are trained and able to exploit the presence of those machines. And on top of that, even when you do go and train those people, there's a massive brain drain problem. They will immediately zoom off to the UK or America or somewhere where they can get 10 times the salary and for the same job. So lots of challenges to overcome. One sort of glimmer of hope around this has been a sort of efforts within the MRI community to try and generate more accessible machines. Accessible in lots of ways. So one of them is cost and maintenance, but also making things that have much less requirements of their users. So Siemens a few years ago now launched this system specifically for this kind of scenario. So this is a relatively low field system. Typical MRI machines are 1.5 or 3 Tesla magnets. This is a 0.5 Tesla magnet. And in addition to being, so that makes it cheaper and smaller, so easier to ship, install and maintain. And has a sort of much more push button kind of interface than the sort of traditional MRI scanners. But it's still the sort of traditional MRI scanner model. It's a big lump of kit that requires a room of its own and is bolted in position in a shielded room. A more radical departure from kind of the standard MRI model is these kinds of systems, which are even an order of magnitude lower field again. So this is a 0.05 Tesla system. As in fact, all three of these things, I've got pictures of 0.05 Tesla systems. But the big difference being that you can see that it's got wheels on it. So it's something that doesn't have to sit in its own shielded room. It's something that instead you can move around, in theory, take it to the patient rather than taking the patient to the machine. And again, designed in a very push-button way. So this is a system, a high-flying system that came on the market. First system like this that was sort of really available came on the market maybe 3 or 4 years ago. And lots of other sort of developments now emerging, trying to create machines like this that are cheap, portable, accessible, and easy to maintain. So that's the dream. But there are big challenges with things like this. If you reduce the magnetic field strength by a couple of orders of magnitude, there is a downside to that, which is the images are crap compared to what you're used to. And so this is sort of where I come into this thinking a little bit with the idea of image quality transfer, which in a nutshell is the idea of trying to estimate a very high quality image given a low quality grungy image of the same thing by learning a mapping from low quality to high quality using machine learning. That's the basic idea. I came to this idea from a completely different place. I arrived at it because, when was this? Probably 12 years ago now. The consortium in the UK got together to persuade Siemens to build and persuade the EPSRC to fund an enormous MRI scanner that now lives in Cardiff, the Connecton scanner. And the idea of that at the time was to really enhance what we could do with diffusion imaging and microstructure imaging. It's not a particularly strong magnetic field, but it has very strong magnetic field gradients, which makes it sensitive to tissue, much more sensitive to features of tissue microstructure than a standard MRI scan. Anyway, we got the money for this thing, built it under the leadership of Derek Jones, and that machine has been installed and done some amazing things over the last 10 years or so. It produces images like we've never seen before. But what I was thinking about at the time was, despite this sort of one standalone machine producing these amazing, unique images, the clinical impact of a machine like that is kind of limited because there's no way you're ever going to put one in every single hospital. It costs tens of millions. It's got the power consumption of a nuclear submarine, and Siemens hate building them because it's a pain and they keep breaking down. But what if we could kind of capture the essence of these very high quality images in some kind of machine learning model in a way that we could enhance images that we get from more standard scanners with the information gained from a collection of images from this one-off super scanner. And that's what led to this idea of image quality transfer, learning that mapping from standard off-the-shelf image to estimating the image we would have got if we'd taken that same person and put them in the super scanner in Cardiff. Very, very simple starting point for this was just a simple patch regression for that problem. So you take a collection of images, pairs of images, low quality and high quality, pick corresponding patches in both, and so you have a set of little image patches corresponding high quality, low quality, and you just learn a mapping from one to the other. Very straightforward idea. You can learn that mapping. In fact, you can do a global linear transformation, or various sort of non-linear transformations that models that you might use to capture that mapping. This was sort of pre-deep learning, as you can see at the time, so using some old school techniques. Very simple implementation of that idea. But actually, even that works pretty well. And you can get some quite dramatic enhancements of images where you reveal some kind of fine tendrils of white matter here, and expose some little fine striations, which is this one. These little sort of stripy patterns, which you can verify from looking at textbooks of these different anatomical regions in the brain. So it produces some quite nice and convincing output. Did eventually catch up with the deep learning revolution, and had various students looking at using those ideas to improve the implementation of this idea. It makes a massive difference. Actually, you get much better estimates of high quality images from standard off the shelf deep learning models. In fact, at the time, the thing we put a lot of effort into was thinking about the uncertainty in those mappings. Where are the algorithms confident in what they're predicting, and where are they less confident? And so Ryu Tanner and Stefano Blumberg spent a lot of time thinking about that. But then back to thinking about sort of low quality, low field scans in Africa. So in fact, this all got kicked off by some may remember the EPSRC or UKRI, I think, had a call for global challenges around about 2017-18. And so we got some funding to adapt these ideas that we developed for the super scanner in Cardiff to enhancing images that come from low field scanners in Africa, which at the time, and in fact still, are, if anything's commonplace, if any MRI is commonplace in Africa, it's this kind of machine. It's a 0.36 Tesla machine, sort of fixed open magnet, permanent magnet system. They're sort of 1980s technology, really, but things that can be produced and maintained reasonably easily. So when you do find MRI scanners in these places, it's usually that kind of thing. But usual problems with a low field system, the images are low contrast and low resolution. So yeah, usual question: can we take images acquired in individuals on a system like that and estimate the images we would have got if we'd taken that patient, plugged them to London, and put them in a 3 Tesla system? So this is a team I'm teamed up with. This was the scanner room in Ibadan, Nigeria. You can just see that open system through the window there. Yes, treated very nicely on various visits there. Again, works amazingly well. So here is some sort of side-by-side, fairly low resolution and low contrast images from the 0.36T system. These are kind of gold standard 1.5T images from the same individual, and these are the kinds of image that we can recover through this learning process using very similar kind of patch-based regression with fairly simple deep learning models. So, qualitatively looks reasonably good. The motivating clinical application of this at the time was detecting and classifying epileptogenic lesions in kids. So quite often you get ambiguous cases where there are smallish lesions causing an epilepsy, and the clinicians aren't confident enough, given these low resolution images, to be able to say that some kind of surgical intervention is worthwhile and what exactly that surgical intervention should be. So the motivation was to try and increase the conspicuity of sort of covert lesions like this one. You can sort of just about see it there, but through this enhancement process, you can increase the visibility of these lesions and help people make decisions on what treatment to follow. This is a somewhat quantitative analysis of the how much this enhancement helps a radiologist to report on one of these images. It's a slightly mixed story, really. So there are certain things sorry, if I flip back to one of the examples of outlining and measuring the volume of a region in one of these sagittal or whatever the other view is, I can't remember. We'll go with sagittal. The sagittal view, then it actually helps quite a lot to have this increase in image resolution. Whereas if you're looking at something in the axial view here, then it tends not to really make a huge amount of difference. But nevertheless, I think that analysis of getting various radiologists to report on these enhanced images, it did show some potential advantages. Moving on to the super low field systems, so this the 0.05 Tesla machine, various other similar types of thing. So yeah, again, same problem. What can we enhance the really quite grungy images that you get from these systems into something that has a lot more clinical utility? So on the face of it, you can find some very nice examples where you feed an image like that into one of these models and produce something that looks like the kind of image that you would get from a high field system. So this is a nice cherry-picked example to show that it's kind of possible to get something sensible out. But it does start to reveal some quite big challenges with this kind of idea. So here's one example of taking a low field image. This is actually one of the ones from Nigeria, one of the epilepsy cases where there's quite an obvious lesion here on the whatever side this is, left-hand side, where with one particular algorithm like this is again cherry-pick this to highlight the dangers. That lesion gets completely obliterated because the algorithm hasn't seen anything like that in its training set, so it just sort of fills it in with the sort of stuff it has seen. which is normal, healthy bits of white and grey matter. So it completely blasts that lesion. Might be okay. In fact, what this algorithm was designed to do was not enhance things for humans to look at them. It was designed to change the appearance so that images would pass seamlessly through an image processing pipeline, through FreeSurfer or whatever it is they want to use. So with that objective in mind, this is not a completely unreasonable thing to do. But it does highlight the danger that if you do want to enhance something for a human to look at it, you need to make sure that whatever you've produced remains faithful to the original data and isn't introducing some, crazy artifact or misleading image content. And this, I think the more that people have experimented with this kind of technique, the more you spot these kind of hallucinations appearing in lots of different contexts. This is one of the other machines led by a guy called Ed Wu in Hong Kong, who's been developing quite a similar system. Again, it's a 0.05 Tesla system. I went to visit them, when was that? Last year sometime, and within 2 minutes of walking through the door, they bundled me into this machine, and this was the output. So this is me here. But this is the raw output that comes out of that system. Quite grungy looking images, and they've actually worked incredibly hard on image quality transfer algorithm, very specifically for their system, and it produces some incredible looking images. You never really think that these have come out of a 0.05 Tesla system, but there they are. But actually, again, if you drill into it, it's not. You don't have to search for very long before you start finding examples of where this algorithm is introducing things which are very clearly not features that are present in a gold standard image of the same subject at a higher field. So, I think that's the kind of challenge here, really. There's clearly some advantages, quite provable advantages in this kind of trick. But there are some potential flaws that we really need to get a handle on in some way, whether it's solve or mitigate through usage or education, whatever. That is a problem that needs to be dealt with. So something that we've been thinking about over more recent years. This is some. I'll just show you a couple of slides from a student of mine, Sungwoo Kim, who's been thinking specifically about this problem of hallucinations. He's done some quite interesting work on this. I haven't really solved it, but he actually started by thinking what he wanted to do was use some of the latest and greatest, deep learning models for this problem. So he jumped straight in with looking at diffusion models. These are going to be brilliant for image quality transfer, way better than the things you've used before. And in some ways they are, of course, but they actually expose you even more to these kinds of problems. And so sort of with a bit of effort, I managed to channel him into thinking about that problem, under the hypothesis that maybe actually, yes, these more sophisticated models can do some very great things, but actually it depends on how you evaluate the output. Maybe if you evaluate the output based on propensity for hallucination, actually my simple linear regression with 3 by 3 by 3 cubes is the best way to go. It might well be that doesn't hallucinate because it's too simple. Whereas his diffusion model will create all sorts of problems there. Anyway, he's thinking about that, and here are some nice thoughts from that. So one observation came up quite quickly is that when hallucinations usually arise when the input data is kind of out of distribution, it's not stuff that the model has been trained on. So this sort of exemplifies that bit. I won't pick through that. But one possible way then of avoiding those hallucinations is to treat areas that are out of distribution differently to the way that you treat bits that are in distribution. So this is basically just a simple demonstration of that idea. All he's doing is to sort of start the diffusion process much later for areas that are out of distribution, so you give it much less opportunity to create a weird hallucination than you would if you started the process from complete white noise. And actually that kind of works quite well. This example is kind of revealing a very simple texture here. for the most part. But so this is a real image that we're asking the system to enhance. This isn't an MRI, something else. But yeah, for the most part, regular texture, apart from some areas where that texture gets a little bit disrupted. But if you train a model, a simple model of this, it will latch immediately onto the idea that there's an obvious texture here. That's what the whole image is made of. And if you try and enhance it, it blasts all of these little glitches, which are all they're actually real. So a standard diffusion model will do exactly that. But if you take this sort of simple trick of just starting the diffusion model in areas that look out of distribution, then it will retain these kinds of feature. And that also works quite well for things like lesions that appear in brain scans. So one simple idea. The other thing, which you might laugh at me for not having done much sooner, is just to make sure that the output of this enhancement process remains faithful to the input. And if you kind of shove an image through a UNet or whatever, actually you don't ask it to do that. But if you frame this whole problem with a sort of more inverse problems kind of framework, then why not do that? My excuse for not doing that is that actually, in order to make that work, you need a decent forward model of what takes you from the high quality image to the low quality image, and we don't have that. It's not obvious actually what a low quality image from a low field scanner that corresponds to an image, high field image that you happen to have looks like. That mapping is not straightforward, which is why they hadn't really done it before. But actually, strength of the naive. So assume we just plug this idea straight into a fairly standard inverse solving framework using diffusion models. It's called diffusion posterior sampling. And actually, even though we don't have a very accurate forward model, it turns out that even a pretty crude forward model actually really helps anyway with this kind of problem by clamping it down and avoiding hallucinations that arise that are not compatible with the, very clearly not compatible with the input data. This is something that's simplifying this year. It's a little bit more than that, to be fair to him. One of the problems with this is that using a diffusion model within this inverse problems context is incredibly slow. So he's worked very hard to find sort of tricks to make this computationally viable, and that's what this paper is about. So that does quite a nice job of avoiding some of the hallucinations that you get from a standard sort of simple unit type approach. That's sort of, there's other things, but that's about all I'll say about image quality transfer, kind of where we are with it. I think, so avoiding hallucinations, and I think thinking about this inverse problems framework, really where I'd like to get to is building this whole idea into the image reconstruction from the raw case-based data that comes out of the scanner, rather than reconstructing slightly grungy image and attempting to enhance it there. But work continues on that. You can pause for questions at this point if you like. That's sort of a natural break point.
We can do questions at the end.
Okay, I'll carry on. How are we doing? Let me change gears completely. I'll talk about some disease progression modelling work, just to give you a bit of background on what this is all about. So it's about trying to reconstruct pictures like this, but from large piles of slightly grungy patient data. Where is this picture? This was a hypothetical picture of how the sort of journey that a typical patient undergoes through Alzheimer's disease from beginning to end. So some of the very earliest signs are things like protein deposits in the brain, which you can pick up from either PET imaging or from CSF analysis from lumbar puncture. Downstream, those toxic proteins start to attack some of the brain cells, and eventually you can see that as morphological changes with an MRI. There's grey matter regions, for example, start to shrink because the brain cells are dying. And then gradually, because of that brain damage, people start to lose cognitive abilities and growth those personality changes towards the end of the disease. So that kind of makes sense as a high level view, and this is despite the sort of intuitive clarity of that. Ultimately, this is a model that's come out of the imagination of, albeit a very well-informed neurologist or neurology community. It's not something that's come directly out of measured data. So what I guess it was sort of prompted by around about sort of late noughties, early 2010s, the neurology community, international community, getting together and collecting some very large data sets of patient data from people with or at risk of Alzheimer's disease. But at the same time, I think that inspired then a bunch of more computational people like me to start thinking about, well, could we learn pictures like this more directly from data as a sort of validation, or potentially an enhancement of these kind of high-level pictures that are coming out from human experts. So quite a menagerie of models came out around about that time. I'll just talk about the one that we started with, a thing called the event-based model. It's really the sort of simplest thing that you could think about for reconstructing a longitudinal picture of disease. Actually, sorry, one thing I should say on the previous slide. It would be relatively easy to construct a picture like this if you had data from a whole load of patients who started at early stages, and you sort of follow them month by month over the decades it takes for this disease to play out. But of course, we never have that data because you can't find people at the beginning for a start, and even if you could, following them that closely over that period of time is just not practical. But what we do often have is more sort of snapshots of individuals at some unknown position along this timeline as they take part in some kind of clinical trial or clinical study or pass through a dementia clinic. So the challenge there is, given that kind of set of cross-sectional snapshots at unknown positions along this timeline, can you reconstruct a whole picture of what this timeline looks like? So that's the challenge. The event-based model is a super simple thing. It identifies a number of different events that might occur along that timeline, and those are typically some marker of disease moving from an ostensibly normal level to something that's detectably abnormal. And so then by fitting this model to a collection of data, the output is an ordering of those events or measurements that you take from a patient, ordered by which ones happen first, typically second, third, fourth, so on. So a couple of examples here. This is from the first event-based model paper, where the measurements were all brain volumes. So we're trying to find the order in which brain volumes start to show abnormality in two different cohorts: some familial Alzheimer's disease patients and some Huntington's patients. And so you get this nice picture of which brain regions show abnormality first and which is spared till the end. And gratifyingly, it shows kind of memory type stuff in Alzheimer's early on, sparing motor stuff until late stages, and sort of the opposite pattern in Huntington's, where you see motor problems early on, reflecting the symptoms of Huntington, and some of the memory stuff sparing later. You also get these nice pictures out of the model that show the confidence in the ordering of the events. This next slide's slightly out of date, but it's a good analogy for what those pictures mean. This is for the football fans out there. A few weeks before the end of the season, one of these sort of probabilistic maps saying the likelihood of each team finishing in each possible position in the league. And that's exactly what these pictures here are like. For each of those brain regions, what's the likelihood that they are at position 1, 2, 3, and so on in that sort of ordering of different events. So they give you a nice picture of which ones, which bits of that ordering the model is confident in. So here the first 3, it's pretty sure they're in that order. But then there's a block of 5 that could be, interchangeable, but we know that they're all before this big block of 10. So that's the EBM, and it caught on quite nicely, this idea. not limited to imaging by any means. So this was really the first sort of usage of that went back to those hypothetical models, which, with lots of, are informed by lots of different kinds of measurement, different sorts of imaging, cognitive tests, and sort of put all of these measurements from lots of different sources on a common timeline. And so, yeah, that was sort of broadly, slightly disappointingly, basically confirming that the models that the neurologists had come up with were pretty much what we saw from the data. But anyway. Okay, yeah, and then lots of other applications of that idea emerged from that point onwards. One of the big simplifications or sort of assumptions that the EBM makes is that everyone is on the same journey. That's how it works. That's how it's able to pull out a kind of longitudinal picture from single time point snapshots of lots of individuals. It has to make that assumption, but it's a big assumption, and it's probably not right. So something that we were working right from the start was to try and relax that assumption and find ways that we could accommodate heterogeneity in that sequence of events or the longitudinal trajectory. And so that was where the idea of an algorithm called sustained subtype stage inference came from. So the model underlying sustain then is that the selection of people that contribute to one of these data sets are drawn from some relatively small number of different groups where each group undergoes a different trajectory of change. And so they're sort of, they define different subtypes of the disease where the trajectory of accumulation of pathology is different in some way. Same data is input, but the challenge now is larger. We're simultaneously trying to estimate what are those groups, what is the assignment of patients to the groups, and what is the trajectory that defines each of those groups. So simultaneously trying to do this kind of clustering into groups and identifying the thing that defines that, each individual cluster. I won't pick through the algorithm, but just show you some output of it, kind of thing it produces. So this is quite a nice example of a validation of that algorithm in it's quite a rare situation where we know that there are distinct groups that have a distinct trajectory of change. So we're looking at genetically defined frontotemporal dementia, where there are 3 genetic mutations that can cause that disease, but they manifest in slightly different ways. So you can analyse the 3 groups individually to figure out what that pattern of change is. We didn't do that here, took the labels away and chucked all of them into the sustain algorithm to see if it would recover those same groups and the trajectories that define them. Actually, it comes up 4, which was slightly worrying to begin with, until you drill in to see who gets put into these 4 groups. And it actually turns out that the first 2 do map quite closely to 2 of these genetic mutations, whereas the third and fourth kind of split the last one, the CMIC group, into 2, which actually we decided was kind of reasonable in the end, because this is quite a heterogeneous group anyway. So splitting that into 2 groups seemed okay. More interestingly, now using that same idea to look at an Alzheimer's disease cohort, where there is no real sort of ground truth as to what set of subtypes there might be. But this is what sustain produces, so it finds 3 different subtypes now, just looking at the AGNI data set. No ground truth really to verify this, but anecdotally, the sort of patterns at the end stages of these 3 groups quite nicely reflect different observations that from post-mortem pathology of different sort of patterns of deposition of neurofibrillary tangles, for example. So there's evidence at least that these things kind of match what's really going on. Okay, let's skip that one. Yeah, and again, I think sustain then as from those beginnings has then sort of found a lot of applications in lots of similar kind of conditions, revealing these different groups of people who manifest a disease in different ways. I picked on this one particular example with multiple sclerosis, looking at this kind of neurodegenerative component of multiple sclerosis, because it exemplifies the potential of this idea, which, if you can find these different groups where that trajectory of change is different, it suggests there's something different in the underlying disease biology among those groups. And maybe different treatments, are more appropriate for those different groups. And, it may cut through some of the problems, the sort of chronic problems in finding treatments for things like Alzheimer's disease, where, it's one of the big challenges they face in clinical trials is that the people are so heterogeneous, and even if a treatment works on a subset of the people in a cohort, that won't show up because it gets diluted by the statistics over a whole heterogeneous group. So this study kind of highlights that quite nicely by showing that for a particular treatment, let me get this right. So one of the three subtypes that Sustain finds for this neurodegenerative component of MS does not respond to a particular treatment at all, whereas the other two, one of them responds moderately and one of them actually responds pretty strongly. Whereas if you take the kind of traditional classifications of MS, primary progressive, secondary progressive, and so on, you don't get any distinction at all like this. So, it's one nice example where it shows that potentially this really can inform that kind of treatment assignment. So a few, I had to show this. Lots of other, kind of advances on the basic modelling. I had to pick a local example. So Peter's done some great work actually on sort of refinements of the EBM and getting it to pull out other bits of information that it doesn't naturally show. So one of those being it will produce a nice ordering of events, but it doesn't tell you anything about how long it takes to go from, between the different consecutive events. So, he's had a very nice formulation to extend it to be able to do that. And another limitation of it is that it doesn't scale very well to numbers of measurements. In fact, I forget what the order is, but it's not quite exponential, is it? But it's a lot, n factorial or something. Whereas, very nice formulation. Again, Peter Taylor published at Neurex last year, cut through that complexity, enabling the model to scale up to having a very large number of measurements put in it. For example, every single pixel of the image. So you could now treat every pixel as a single event and produce a sort of video of how these, how a disease progresses pixel by pixel across the brain. Last topic, how much time have I got? Okay, I'll try and be quick on this one. So the EBM and Sustain, do some interesting things, but what they do is sort of pull out patterns from data sets. They don't explain what those patterns mean or where they come from. And actually my original motivation for developing some of those early algorithms was much more along the lines of trying to test hypotheses on what are the underlying processes that cause the sort of spreading patterns of pathology that we see in these conditions. And the reason I sort of went back to think about that ordering was I kind of thought, well, you can't really test any hypotheses on what mechanisms are causing these changes over time unless you can get a picture of what the pattern of change over time is. Anyway, sort of having developed some technology to do that, and then started thinking more about what are the possible mechanisms that explain these patterns that we observe. So lots of hypotheses out there about what causes the particular pattern of pathology aggregation across the brain in Alzheimer's disease, other neurodegenerative conditions. For example, could it be that bits of the brain that are very heavily used are most vulnerable to this kind of pathology? Or could it be the opposite? Bits that you don't get used very often. So this is the first one, like a sort of wear and tear type hypothesis. Second one, the opposite. You use it or lose it kind of thing. If you don't use if a bit of the brain isn't used very much, it's more likely to be attacked by some kind of protein. Another one, maybe the pathology starts somewhere for some reason and then crawls along the connections of the brain, the white matter that connects it to other bits and spreads along the sort of connectivity substrate. Or maybe it's a similar thing, it just sort of spreads through proximal diffusion. So anyway, lots of possible hypotheses that people argue about with, have biological arguments for. But I think, having got a picture of how these things evolve over time, you can start to, these things are quite simple to encode computationally, and you can compare the predictions of a simple computational model of these different mechanisms to the sorts of patterns that you get out of sustain or out of the EBM. So, yeah, this is something I've been working with a little team on for a few years now. I won't go into too much detail of the keep it for too long, but one of the challenges that you immediately face is, so I think probably that there's quite strong evidence that the connectivity of the brain plays at least some role in the way that the pathology propagates. And so, fine, we can build models that capture that, but they rely on having some picture of what the brain connectivity is. And those pictures of brain connectivity typically come from quite crude sources, things like diffusion MRI tractography or resting state fMRI, that give you some picture of connectivity, but they make a lot of mistakes. They're quite crude and they're very uncertain. So, can you really discount, a particular model based on functional connectivity because it doesn't make a good prediction when you can't really rely on the picture of connectivity you've got? So we've been thinking about that a lot. First of all, what are the best sort of pictures of connectivity that we can generate that feed into these models? And are there ways that we can mitigate the sort of false positive or false negative connections that come out of these slightly crude estimates? So a very nice bit of work from Ellie Thompson last year, where she was evaluating the viability of these various models, given different views of connectivity, first of all, but then also thinking about, okay, are there ways that you could combine different types of connectivity in a way that they best inform, they best predict the observed spreading of pathology in, say, Alzheimer's disease? And it's led to some quite interesting thinking about connectivity. And, in fact, can you even do the opposite? Can you potentially sort of fix errors in predictions of brain connectivity by thinking about the connectivity pattern that best predicts the, pathology spreading patterns that we observe in Alzheimer's disease? And can just make it part of the whole model estimation. Another interesting thing, can we bring the whole of kind of history of publications about brain connectivity to bear on this problem and sort of fix estimates of connectivity based on what an LLM tells us? Can an LLM, what does it tell us about the likelihood of connectivity between region A and region B? Actually, quite a lot, it turns out. And so this is a nice bit of work that Ellie's just submitted for review, thinking about kind of fixing connector models using information contained in an LLM, and actually you can get some quite significant improvements in the prediction of these pathology propagation patterns if you correct by anatomical knowledge contained within an LLM. I'll skip that one, similar kind of thing, and just finish on this one, which is kind of I think sums up the effort that we're trying to make, on this tack at the moment, which is, given all these different views of things that are relevant to predicting these patterns of pathology propagation and understanding the mechanisms, how do we build a big inference engine that can understand the uncertainty and errors in an estimate of connectivity, the uncertainty that's contained in image segmentation, the sort of grunginess of the models themselves? Can you take all of that into account into an inference engine that will then evaluate the evidence for a whole, for a bunch of different kinds of model? That's sort of where we're trying to get to with this. And on that note, I shall stop.
Very much, David. Great talk. So, questions as well. I'll keep it short in the interest of time, and students first. So,
thanks very much for the very cool. I had a question about the image consequence or problems. We thought about this as well in terms of diffusion theories happening, and you focus only on average distribution hallucinations, or the real bigger problem about age distribution hallucinations?
Yeah.
Right, so have you thought about how you tackle those?
Yeah, so we've been, I think our terminology has been intrinsic versus extrinsic. So where, I forget which way around it is, but one of them is a hallucination that is not consistent with the input data, whereas obviously there's a whole other null space of things which are consistent with the input data, and in a way, those are much harder things to tackle. I don't have a solution to that, unfortunately. I'd love to, but hang on. I know Shumhoy was telling me he did think he had a solution to this the other day. I can't remember what it was, and I probably shouldn't tell him it. So, but the short answer is no. I don't know how to solve that.
I've got a good question for you then.
Yeah, definitely.
Any other student questions?
I mean, just I guess to pick up on that. I mean, one thing we can do, of course, I think, is highlight where those things are likely to happen. You can at least try to evaluate, the uncertainty of the output. You'd hope that where those intrinsic or extra, whichever ones they are, where those kinds of hallucinations happen, that you should be able to get the model at least to say there's a high degree of uncertainty in this region.
Thanks. Okay, if there are any other student questions, then Thomas, do you have a question?
Yeah, I wanted to push you a bit harder on the exact same issue.
Yeah.
Whenever I'm asked about this, when people don't know anything about how these things work, which occasionally happens in the press, people also answer, oh, someone enhanced a picture of Donald Trump and he seems to have a love about his ads and something like that. And so then you comment as, say, the outsider. That I have the impression that if you want to improve your holidays, then absolutely want one kind of reality that looks great, then of course you can do whatever you want and they will look great. Now for the medical imaging, you don't want it to look great necessarily. You want it to give you information about an abnormality in the brain. Isn't improving the image with the distribution of normal images as the input going in the opposite direction of what you want to do, even if it looks great afterwards, will erase stuff. So that's why I said about pushing it harder. I feel you're almost going and disimproving the image by making it look great, but with less information than before. What do you think about that?
Yeah, it's a tricky one. I mean, I think the interesting thing is that the consumer of this, the radiologists, they love it. They think it's brilliant, and but they are a bit obsessed with having images look amazing. And actually, you're right. They don't actually need them to look that amazing a lot of the time. And it's actually, one of the things from that study of the epilepsy cases in Africa, one thing that became apparent fairly quickly is that, yes, there are nice examples where that kind of enhancement can reveal a lesion that was very difficult to see otherwise. But it's quite rare that you hit that sort of Goldilocks zone of, where the lesion is just small enough that you can't really make it out on the low field image, but big enough that there is still a signal there. It's quite rare to fall into that bit. When it happens, great. You make it more conspicuous. They spot it. They love it. But most of the time, that it doesn't happen. Where it does make a difference, I think, is sort of feeding into downstream processing. So making an image more like what a downstream processing algorithm is used to seeing is actually super useful, because it means you can take those hyperfine images and you can put them into FreeSurfer and it works. Whereas if you just take them as a raw image and it does not work at all, it completely falls flat. But if you've got a kind of, you want to do some high throughput analysis of images like that to make some sort of standard measurements, it really makes a difference.
It works, but how much? I mean, you showed the synthesis, for example.
Yeah.
When there's that big lesion there, it does something.
Yeah.
And it will give you'll be able to segment it, but then the measurements don't necessarily mean anything in those regions of interest. And if that's not propagated downstream, as you talked about your previous work, you had looked at the uncertainty of these measurements, but did you ever propagate that into a downstream task? You can use it for anything.
Well, so probably, usually the downstream tasks are not built to take that information in, but we could. I mean, so that, the SynthSR example there, was a bit extreme, obviously. But I mean, you can argue that, in that case, if you're going to use free surfer, then you're not really looking for that kind of lesion. What you're after is brain volumes and stuff, and so maybe it's okay, but I know it's dodgy. I'm with you.
Okay. In the interest of time, is there any other questions?
Kind of a naive question, right? But sort of going on Thomas's point, well, it feels like you're not adding in any more information. Is there any move to basically get them to move a little bit, or take some snaps over time? So at least you're combining. You've got more information, right? That's what I would do naively. I lower res the image, take a few images, and move the camera a little bit, and then I'll get high res. Isn't that a better way to go? Is that a way one can go?
Not really, I think, is the answer, because there's always a certain amount of time you have to image someone, and and let's say that's 20 minutes, in a typical clinical scenario. And with those machines like that, that's what you get in 20 minutes. And so there are lots of tricks of acquisition that you can play. The simplest thing is you just repeat it several times. And people do that in order to get the kind of images that we're already seeing. Things like sort of jittering and moving around actually often don't really work. There are things that you can do like that. So, for example, one thing you might do is rather than acquiring one image at a particular resolution, you acquire three, where in each of those three, it's that resolution in plane, but you much fatter resolution across the plane, and you do it in each of the three directions. And then you can try and stitch things together to create them. So there are tricks of acquisition like that. They're sort of complementary, really, to this kind. You could do both, and there's no reason why, if you do that, you couldn't then also do an enhancement that is. So I'd slightly challenge them not adding any new information. In a way, you are, because you're adding the knowledge of what the whole distribution of high resolution brain images look like, and obviously you have to make a guess from within that. Yeah. But yes, there definitely are things you can do from the acquisition and reconstruction point of view. But I still think that there's kind of a place for this somewhere. Maybe not. Maybe it's just too misleading.
Probably last question.
Is there any space for kind of a better forward model, so like physics and slide forward model, to actually have some understanding of what the difference between a large magnet and a small magnet?
Yeah, I think there is actually, because it is very dependent on the forward model, and what we've done is, for the most part, is sort of construct that through example. But you could, build a big sort of block equation model to try to predict what a low field version of a high field image looks like. So people have done that, actually. There are examples of that out there. The problem is they end up not very accurate, because it's often not the basic physics that drives the end appearance. It's a lot of the sort of tricks of acquisition that the radiographer never tells you that they've done. So it's quite hard to capture the full complexity of it.
Okay. I actually have a question, but I'm going to hammer you on IGT. I recognize. It's a more general question. That might be helpful to people, though. More generally, is that you talked about methods today that are in the area of trying to get useful information out of data, basically, coming up with methods that can give extra information, let's say, from data that are available. And what do you think about the methods that you presented to make successful compared to other methods? I mean, that's very broad question, but if it was a flash in the pan, it was just published at the right time in the right place, for example, and everyone latched onto it. But you consistently provided these methods, they've been consistently successful. So there must be more than just.
There's been lots of casualties on the way, by the way. These are just the survivors. But thank you anyway. What made these things survive? I think a lot of it is tapping into things at the right time. So that the disease progression stuff's quite interesting where that came from. I can still remember very vividly the initiation of that whole idea, which was a 5-minute conversation with Nick Fox. Nick Fox is a neurologist, sort of consultant neurologist, and head of the Dementia Research Centre at UCL. And we'd been talking about, the sort of pathology patterns in dementia, and he said, "Oh, it would be great to look at the connectivity of loss." And I said, "Well, what's that?" And so he was talking about this hypothesis that the propagation of pathology in Alzheimer's disease is something to do with brain connectivity, and pathology somehow gets propagated along brain connections. And he said, "Is that something you think you could model, or try and come up with a verification computationally that is one of the mechanisms that's at play in this disease?" And so I sort of started thinking about that. And that, as I kind of said during the talk, that's what led me to think, okay, before you can answer that, you need to have a clear picture of what the temporal order sequence of things is. So, but I think that's kind of why all of these ideas caught on, is because they were tapping into something that actually quite a big community was already thinking about. And you can frame it within that and point to, this is important because it enables us to answer these kind of questions. I don't know if that
depends on what the audience thinks, but as a way like disciplinary research, that's we're all interested in that, particularly Sussex AI.
Same with IQT, really. It got a resurgence because of the trend towards low field MRI.
Right.
I mean, the sort of questions about hallucination and reliability arrived the first time I gave the talk, and I still get the same questions now. And they're still good questions. But nevertheless, it sort of taps into something. People think, that would be really good if it worked. And, yeah.
Great. Okay. Let's thank Danny again.